a chief scientist in CCMB, Hyderabad. She is an expert in molecular biology and biotechnology, contributing significantly to cutting edges of research in the life sciences. Let's welcome Dr. Vishnupriya ma'am with a huge round of applause. Because of what? Plant molecular biology.
Prasad.
Yes, sir.
एक फोटो घेतो सर असू दे अजून एक बटन बंद कर अजून एक बटन बंद कर
Well, good afternoon, everyone. At the outset, I once again thank Rajeshri South Mahavidyalaya, Shiv Chhatrapati Shikshan Samsthan, for giving me this opportunity to interact. And I thank all the dignitaries. All the dignitaries that are on the dais and off the dais, faculty, research scholars, students, teaching and non-teaching staff. A warm welcome to all of us to this national seminar. Well, a hearty congratulations to the team, department, botany, and all the staff and students, technical, non-technical staff who are behind this, organizing this seminar. It's a real task. I know the work must be on for at least a couple of months. And here we are together to share a few thoughts. Excellent. Well, actually to say, I have no points other than what Professor Shinde has mentioned. He has actually covered all the points. And I will actually run through my presentation very quickly. But my main aim would be to motivate you students at the end towards research. So there's nothing new. You have all the platform of Google where you will see that what I have listed. Most of you are aware of. But since I'm told to make a presentation, I put some efforts in consolidating some points together as a presentation. So we'll quickly go through this. But at the end, I would like to lure most of you towards opting the research as your career. So let's put that effort together. We'll just quickly run through my slides. As you can see today, integration of biological sciences and technological innovations are completely revolutionizing the life sciences, as our principal had told. Under the umbrella of life sciences, we are covering almost all topics that are essentially playing a major role in our lives today, rather in the ecosystem. Okay, so let's see what are the challenges that we are facing in healthcare, leading to personalized medicine, agricultural advances, and environmental sustainability. So in the recent years, we have witnessed a lot of changes. For example, what we are all wearing our smart watches, how it has been integrated into our life system and what is the role that is playing. It is actually giving us an information to be very cautious. Almost all of us must be using the gadgets and using the benefit. So what is the outcome of this is we can be preventive, a precautionary measure can be taken. So that is an advancement of healthcare technology that has given us. Next slide. So what is the goal? To enhance the quality of our life through responsible integration of technology and biological sciences. So what is that we have to do? We have to balance our innovations with ethical standards, sustain and develop a sustainable future. So ethical standards, we'll go through them in detail once again. So ethical standards play a very, very major role when we are trying to bring new advances. Next slide. So I have just listed a very few of them here, and we will just very briefly discuss what is revolutionizing healthcare and technology, personalized medicine and genetics, emerging technologies in life sciences, nanotechnology in medicine and biology, sustainable agriculture and food security, environmental applications, ethics and policy in life sciences innovation, ethical implications of augmenting in human capabilities, AI and machine learning, and global changes and the role of biologists. Next slide. So as we discussed, all the wearable devices, what does it do? It is tracking our health matrices like heart rate, footsteps, and sleep patterns. So what happens with this? We become proactive, we become very conscious. So we eat healthy and remain fit, and we should take advantage of this technology innovations. But we should not be take it in a negative manner and be very alert and stress on this. So that is the negative consequences of this technological innovation. So we have to use it very, very responsibly and carefully. So what happens with this? It's an improved access to healthcare services and better health monitoring and timely interventions. So suddenly somebody's BP is shooting or the heart rate is shooting. You can take a very precautionary measure and rush immediately to the required healthcare centers. So this is the major advance I think that has happened in these kind of gadgets. We are all aware of this. The same is explainable in this slide. Next slide. And what's we saw during COVID, all this telemedicine that is happening. We are all going on an online consultation with doctor, which has reached the corners of the country, and all the senior citizens are continuing to utilize this facility. So actually the pandemic has revolutionized our lifestyles, and the technology that emerged with the experts have really benefited worldwide. Right? So how do we think personalized medicine is going to help us? Tailoring medical treatment based on individual genetic profiles, Professor Shinde already mentioned. Now we have the human genome sequence that is released with the efforts over the years of several scientists. And how are we going to utilize that? Is the genomic sequencing. We know of this breast cancer, there's which is called Herceptin. This HER2, it's a positive for breast cancers. So what happens with this use? The protein is produced more and then it detects the cancer cells. So it enables to kill the cancer cells. So that is an advantage which the technology has advanced. But we have to be careful. There are several side effects of this. So without consulting your concerned physician, you cannot take because it leads to, if somebody is having a heart problem, you have to take care to consult a pulmonologist or a cardiologist before you take this because it enhances the heart problem. Similarly, sometimes it causes diarrhea, chills, fever. So a continuous monitoring is required. So with this advancement of technologies, there are side effects that are linked to it. So it has to be used very carefully. So every technology advancements have to be dealt very carefully. Right? So similarly, we have all heard of CRISPR technology, which Dr. Shinde has said. So what is CRISPR technology? It's a palindromic repeat that is there. And how do you do it? There's a Cas9 inside where you incorporate an RNA and it detects, it's a molecular scissor. It goes there exactly and trims the gene that needs to be deleted or inserted. So efficient technology that has come. So as he rightly mentioned, this can be used for to cure lot of. This has been used in treating muscular dystrophy, which is a very serious, devastating, and a very highly prevalent disease. And several rare diseases that is a very ignored area in the country where a lot of funding is being given for R&D towards this rare diseases, where the CRISPR technology will be used very, very efficiently. So it needs a lot of planning, a research planning which has to be done. It has to be executed properly, and here again we have to follow all the ethical protocol standards that we need to, implement while we are using this CRISPR technology. So this can be used in various fields in health, crops, environment, and so many other utilities. So what's happening with this technology? Here I have just listed a few of them where it can very efficiently and it can produce faster results with this technology rather than following a very traditional way of identifying the mutation, deletion and insertion. So this happens very easily. You can insert or knock out the whole gene as such and do these experiments on cell lines and mice is very easy, so we can advance for clinical trials very faster. So that is the advantage of using this latest CRISPR technology. Next slide. So if we come to nanotechnology, most of you are aware from the internet and several research articles. When we say nanotechnology, what is it? It is very nano, right? Very small in size. So when these nanoparticles are used to treat or cancer cells, it very precisely goes and delivers the drug. So there are a lot of already in practice treatments for cancer patients using gold nanoparticles and silver nanoparticles, which makes it very precise. Of course, the cost is one thing which has to be worked out more, and I'm sure scientists and research will all come together. As he said, different fields have to come together here, like industry, academia, and IT people. All of them coming together to make it more cost effective and more efficiently, where all the fields of people come together to work for the benefit of the world. So as he rightly told about bio, a whole lot of data can be analyzed with the help of bio, which is, I think, a major advantage in today's science. You're having enormous big data. So you have, suppose you take plants, you have all your genome sequences that are released and annotated. You know the genes, you know the proteins. So you have to pick up what do you want to do. You want to incorporate agronomic traits. Where are the genes? Which chromosome? Which you can precisely pin the function and the position of the gene, and that can happen only with the help of bioinformatics data analysis, where you can then compare across genomes now. And you all have heard about next generation sequencing, where a genome can be sequenced within few hours and data can be given for a whole genome, properly annotated maybe in less than 8 days. So you can see the advancement of next generation sequencing, where if I tell you about personalized medicine, our director, Dr. Laji Singh's dream 20 years ago, he used to say, no files, no books, your genome will be sequenced. You go with a chip to your doctor, and your chip will be inserted in your doctor's PC, and he will know what is your genome sequence, what are your symptoms for your illness, and he will see to which gene it is responsible, and you will have a prescribed, personalized medicine that would be so effective and so very cost effective also. And treatment would be so effective. And I'm very, very sure that very soon India is going to progress with this kind of personalized medicine, because all of us are working towards this kind of a treatment where I think we don't want to have any side effects. We want to have a very personalized medicine. Rather, we want to take very less medicine to keep ourselves fit. So, with this next generation sequencing and CRISPR technologies and so many antibodies, monoclonal antibodies and nanotechnology and bioinformatics data, I'm sure our lifestyles are going to enhance. And we have all heard of old age beginning at 80. So we are all very young. Next slide. So just switching gears to sustainable agriculture, you have all heard and seen now how drones are being used, and how very nicely everything can be managed, and we are helping the most important person of our country, the farmer, by facilitating him with this kind of a technology, and everything comes on his mobile on an app where he can sit and monitor and take the advice of your village development officers in agriculture and maintain and manage his farms for a better lifestyle with a good revenue. Next slide. So, I will take you to the kind of research that I have done, and our rice research has bagged 2 national awards. Next slide. So, we have developed a rice variety called improved Sama Masuri. That is, it has a very low glycemic index, below 50. So, it releases sugar into the blood very slowly. So, it's a diabetic friendly rice variety. And this rice variety took us about 10 years, 10 long years. So, some farmers had approached a rice research institute then called IIRR at Rajendra Nagar, where a very severe disease, bacterial leaf blight, was almost devastating the whole crop. So DRR scientists and CCME scientists, we all came together, and then we thought, what could we do to the farmer to help him to not lose his crop with this disease? And this is not any genetically modified rice variety. There is a parent variety, ARDLS-2025, that originated in Pantnagar and came to agricultural university called Jaishankar University. But this disease is not a fine slender grain rice variety. One is resistant to the disease, harboring the resistance genes. But we wanted to introgress this into the fine slender grain, medium grain rice variety. Because it's a popular rice variety in and around 5 states of Telangana and Andhra Pradesh. So we only introgressed 3 resistant genes into this variety across 4 generations, and we ensured the presence of resistant genes into this rice variety, retaining the parental characteristics. And to our surprise, when we did the experiments in the greenhouse, it turned totally disease resistant. And that was a joy. Well, it's a very hard journey. It was 18 hours of work. And when we give the free seed to the farmers, when they line up with folded hands This was that's a great honor that we could contribute to the farmers, and they have enhanced their lifestyles because of the higher incomes they get. And this variety had an added advantage of 10-day early crop and 20% more yield.
Yes.
So, and then Governor Narasimhan was not believing us. He said, I want to meet the farmers. So we went to the village. We had a farmers meet like this. Thousands of people. More than maybe 3,000-4,000. And then when he called the farmers and he's asking them what is the variety you're growing, variety was not released. He went to release. He was astonished to see that the farmers are already growing the variety and enjoying the variety. So he was surprised to see that he could reach out the village. Being a molecular biology lab under Council of Scientific and Industrial Research, we collaborated with the Rice Research Institute that comes under ICAR. That was a successful biotechnological story that CSR flagged off, and we got 2 awards, and Modi ji was to give us the award. He addressed the gathering for 1.5 hours, and he had no time left to give us the award because he was heading for another meeting. And then our Science and Technology Minister bestowed the award to us. So I would proudly say that I got an opportunity single-handedly work in this project in collaboration with another rice research lab. So all the breeding of plants was done in the other lab because they had acres of land, and our lab being a small lab, but we have all the latest infrastructure. So we did all the molecular analysis, and we could give out this kind of a wonderful rice variety called improved Sana Masuri, which is a diabetic friendly rice, and now this is on Amazon where people are able to buy. But yes, we have to scale it down on the price because we have to have more farmers to grow it and we are buying back from them. Even today, we give every farmer a 10 kg free breeders rice variety so that he could save from his crop and go for his next crop. They don't have to buy the seed again and again. They can save from their crop and go to use the same rice seed for the next crop. So this is the rice variety, and I would like to tell your students that research is an amazing journey. We didn't begin this work expecting that we will come out with a disease-resistant rice variety. We sincerely decided just to see and help the farmer. But when you put your honest and sincere efforts, you will see that the outcome is very beautiful. So research is a very enjoyable journey. And I think when you all plan to take up the career, you will enjoy. Even if the experiment fails today, you will come home very happily tomorrow. I have to set up the PCR. Why didn't it work today? Shall I change this? Shall I try that combination? What is it that I have to do? So they are the thoughts that will be going in your mind all throughout the night. And when you fall into that kind of a path in your thoughts with your research activity, I am sure you all will bag some awards for this great institute that you are studying. And we all look forward that you all will fall, most of you, I would say all of you, will opt to go into the research activities. So these are the benefits, and you can see that this is a rice field that is severely affected with disease. It's in the same field as this is the improved Sana Masuri that's totally free of the disease. Next slide. So these are the estimates that the farmers have, brought it out, and very huge. Now across the country, we have popularized our rice variety, and farmers across the country are growing it, especially in the state of Uttar Pradesh. It's a very, very, the farmers are so very happy. I was there in 2016 in Kisan Mela in the lab there at CMAC. I had introduced it there, and till date, at least I will get 1 or 2 calls every day asking, मैम कैसे हैं? हमारा फसल बहुत अच्छे से चल रहा है, बहुत अच्छा फसल हुआ है. So it gives them immense satisfaction and joy because it has changed their lifestyles. So they wait to they see. Even this season, we have sent them so many seeds. Next slide. So that way here you can see in the same rice variety, we are trying to develop more disease resistant to blast, sheath blight, yellow stem borer, and early maturation, high yield, low phosphorus, drought tolerant, and so many soil salinity tolerant. And most of them are in trials. Probably this year we will be releasing a few more varieties that will come up with low phosphorus and high yield. They've almost passed the trials. I think before 6 months we'll be releasing some more varieties. Next. So there are some examples where you can see that the high yield varieties that are in this panel. And so you can see actually Honorable Prime Minister, he had announced these varieties in his speech, and it was really an honor for us and a surprise for us when he called them out. So as I told you, high yield and for blast and for garbage and salt tolerance, they are some now 1 year back pictures there. They have all passed the field trials at different locations of the country, and we're just making the formalities to release. Next slide. So, what are the future plans? Trials work will be to make more products having the abiotic stress, yield related and nutrition related, and all these resilient crops. So all our efforts are being nurtured into developing tolerance to all these stress. And just giving you some numbers of what I told you, not very important. So again, we can see what's happening in the environment applications. I think we will have our speaker today, Avnashi, who will give us more insights on bioplastic degradation and environment sustainability. So I think I should not be touching on this point. It's like this. So ethics, it's very important. So why do you think genetically modified foods are having such a resistance? Because ethical protocols and standards have to be very strictly followed. So the fear of genetically modified foods or whatever comes because maybe the protocol is not followed. So that's the reason the resistance and use of genetically modified organisms or plants or foods is still prevailing across the globe. So each one of us has to be very responsible in following ethics, be it experiments or be it in our life. So we have to have a self check and we have to have our protocols set and we have to follow them in whatever research activities we are making. And I think policy administrators have to make a very strong policies that all of us, each one of us, have to very strictly abide by these policies that will be framed for research activities. Next. So, and this actually, Professor Shinde had touched design of babies. What are we going to do with that? Next slide. What happens in the design of babies? Like, you are picking up traits, as he said, for eye, hair, height, whatever reasons, intelligence. So these genes, if you're putting into the babies, well, that's fine. But what aspects of those genes are still not uncovered? So that is the greatest danger when you think of these designer babies. And then, as he also said, what happens there will be a homogeneous population, there will not be any diversity. So what happens? A rise of genetic diseases, rare diseases will happen. So while we are enhancing our technological advancements, we are also equally rising the disease probabilities, which will be very, very dangerous. So there again, ethics will play a very important role. So I think still rules and guidelines are being framed for all these kinds of technologies that are being advanced. They are not fully in place. And so I think once all such guidelines and protocols will be in process, things will, be in a better shape. Artificial intelligence. Well, we are all in the boom of artificial intelligence and robotics. Don't fear that you will be replaced with artificial intelligence. Human empathy and human touch can never be replaced. Well, but we have to be very cautious. We also have to equip ourselves with the artificial intelligence skill. If we do not, somebody else, my contemporary scientist with an artificial intelligence skill will surely display. So I think that is a challenge to the younger generation that what are all the programs that you need to learn additionally with whatever you're doing to skill up yourself to get your concepts very clear. I feel be it scientist, be a medical doctor, be a professional, a businessman, across the fields, as our honorable chairman said, we have to go on learning, go on equipping ourselves. Only then we will sustain. Otherwise, surely we'll be dismissed. So AI is it's a trigger for us to keep ourselves updated and continue our learning. So this is what it is. It will be very helpful again, with the view of bioinformatics, where it will help us to predict and take a timely decision. So that is a major change that we all are going to have. And with the help of robotics in the medical field, in agriculture, and so many other things, AI is going to really sweep out all the fields across the globe. So these are some examples of AI influence in surgeries, in delivering medicines through nanotechnology, very precise and very quick, effective measures will be there. Next slide. So what are the global challenges? How the fields of biology and technology can collaboratively address these pressing global issues? For example, the COVID-19 vaccine story which we have heard. So we know there were so many different kinds of vaccines. It was attenuated virus, attenuated different virus, and the spike protein. All these came out with different names. Covishield, Covaxin, and the Johnson and Johnson vaccine. So many things. So different types of vaccines came. But CCMB has taken up a very different view of it. So whenever there is a pandemic, there has to be, you shouldn't begin from the scratch. So we have got a very major project. We are developing an mRNA vaccine platform where a platform will be ready. Whatever the pandemic or the virus comes, you just take from there and just specifically modify that particular protein, whatever is being expressed or by these viruses on the cell membrane. So we have just initiated this project, and I think CCMB will again render a major help in this kind of pandemic breaks. And when we saw that all our Swiss had 37 labs, how we all came together and we made different kinds of products to combat the COVID-19. Well, we made kits, we made PPE kits, and IICT, our sister lab, played a very major role in a component which Bharat Biotech released the vaccine which sir was mentioning. So IICT, our sister lab, a CSIR organization, played a very, very major role in that vaccine deployment. And CCMB as such, we played a very major role in many, many activities of COVID-19, and especially the healthcare counseling, where our director used to give a lot of media and press releases, which gave a lot of moral support across the country. And not only just moral support, but we were into several products that were combating COVID-19 as a whole organization of CSR with all our 37 laboratories. So our strengthened health systems, improved public health outcomes through biotechnological inventions and discoveries will definitely transform our lives. So of late, very recent paper in Nature about anti-aging, where we can say that as it's at 80. So what happens? So if you make the change in the nucleus, the longevity increases. But then again, ethics come and play here. What about the resources? What about the population, blowout? How are we going to sustain it? So what are the ethics? What are the standards that are set to follow? Are we all going to say yes to this or no to this? So this is a big debate when the standards are being worked out, and I'm sure in the coming papers in Nature, we will be seeing some more exciting results. So, we all may become immortal, we don't know. Next slide, please. So, these ethical dilemmas remain. What happens? We all extend our lifespans. How are we going to face the standards and how are we going to sustain for our necessities? It's a big challenge. Yes, the longevity is going to increase, but we have to balance our other requirements, without which only longevity increase will not be very beneficial, will not be a happy scenario. Okay? So the convergence of technology and really understand by this is the integration of biology and technology holds a very, very transformative potential, but the importance of ethical practices and responsible innovations for promoting sustainability. If we cannot sustain something which we have found out or delivered, we are going to perish. So sustainability is again a very, very important point that we should keep in mind and we should keep on evolving on. So what do we all have to do? So people across the fields of science or business, industry and academia, we all have to come together to navigate the future of life sciences responsibly. And I'm sure we are all going to come together for great changes that we are all expecting to enhance and get incorporated in our lifestyles. So if this is with this presentation, I would like to know how many of you all would be interested in going and taking up your PhDs as your research careers. Please raise your hands. Oh, I will tell the people who didn't raise their hands do not want to enjoy life. Because I'll tell you why. Government gives you a fellowship of 45,000 rupees per month if you pass an exam called CSIR UTC NET exam, which is held twice a year, once in June and once in December. I'm sure each one of you sitting here is above average. If not, you wouldn't be studying in this sanskar. Anybody below average? Please raise your hands. Everybody is entitled to do PhD there. Nobody is below average here. How can anybody be below average in this sanskar? Not possible. So it's a very simple exam. I'll just take few more minutes. Exam will comprise of 12 chapters across your subjects. Please download the syllabus and only pick up 8 chapters that are interesting to you. Enjoy learning those 8 chapters thoroughly. Solve your back year's question papers. Get a feel of the exam. Take some online exams. And I'm sure each one of you who attends this exam will surely come through this CSR exam and bag a fellowship of 45,000 rupees, and you will demand your sanstha to improve more on facilities and do research here in this sanstha. I look forward each one of you to please today look at this website of CSR the and please see the papers. Last. Next. No, last letter. Okay. Do not hesitate to ping me if it is for this exam. Unless I'm in a meeting, I may not take your call. But I would be glad to discuss with you on WhatsApp, on a call, anytime. You're welcome. So as Professor Shinde had pointed out, I don't know the number in this call. A handful of you will surely, I want all of you to do your PhD, of course, but a handful of you will surely get awards and laurels and prizes in your research activity with great outcomes and international papers. And this is my trust and faith after seeing all your eminent faculty and members and the facilities here. I urge each one of you to please take a look at this. If you have doubts, please ping me, call me, and I wish all the best and great success to each one of you in your careers. And to conclude, I will once again thank Professor Shinde ji for giving me this opportunity. And Chairman Sir ji is being so patient to me. I thank all the faculty, students, research scholars, and all the eminent dignitaries of the dais and on the dais for patiently hearing. Thank you so very much.
That was truly inspirational work of yours, ma'am. It is really thought-provoking for us, and I will let you give the word of mine. Me and all the fellows and students definitely doing enters in the area of research field definitely, and we will let you know, and we will comes in contact with you, and definitely do some research and show you, sir, ma'am. This is our institution already has provided many research facilities, as well as our syllabus has also developed as per the CSIR way. Thank you so much for your words, ma'am. Really so excellent. Is anybody has a question? Session is open for the question and answer.
If students have any question, yes, please.
Hello, ma'am. I'm a student of microbiology. It's my question that cancer is due to the mutation in the gene. So it is related to the gene that it will transfer in the next generation if woman or man is suffering from
That's a good question. Cancer is related to a specific gene, you're right, but it's not so simple that it only restricts to a gene. It's a very complicated interesting pathway where several genes will be playing a role in it. So it's not that it's just one gene. And your question about whether it gets transferred to the next generation. See, we all know at the end of 21 days, the cell dies, right? And the cancer cell never dies. Cancer cell never dies. So what happens? It goes on multiplying. But if suppose there is a chemotherapy or surgery is done, and one cell escapes in the host, it remains. It reaches another destination within the body. So eradication of each and every cancer cell is very important. So your answer to the generation, even if one cell escapes, the disease relapses.
Thank you, ma'am. My next question is that this is the summer season and we all are enjoying with the watermelon. And this news has spread on the social media that the watermelon, which has the largest size, it is due to the insertion of the injection to increase the cell mass of that watermelon. So it causes the suppression of the tumor suppressor genes and it activates to the cancer or oncogenes.
See, that's not. So, whenever there is a tumor, there is a suppressor gene and there is an oncogene, right? So, what happens when the oncogene takes over the suppressor gene? The cancer cycle goes on and on. So, it's a fight continuously between the oncogene and the suppressor. So, unless the suppressor gene is able to combat the oncogene fully, the disease will not be curable. So I'm not saying that cancer is not curable, but there are early markers. Early detection is possible now via markers at a very early stage, and you don't even have to have a physical, extraction of the tumor or the tissue. It's a liquid biopsy which is happening with a very early detection. So that's why a regular checkup and a prevention and a precaution has to be taken each time. You don't wait for the tumor to develop. So there are symptoms where a patient feels chill, fever, weakness, pain in the muscle. So don't neglect. Get yourself checked. It need not be a tumor. It can be benign. It can be something else. So health symptoms when they grow up should never be neglected. You should always rule that out.
Thank you, ma'am.
Hello, ma'am.
Sure,
Brother, I think you
sure,
have
sure.
developed a Sangama story. Which mass selection or pedigree selection, which type is used by you for the development?
We use microsatellite markers across the chromosome. And these were two dominant resistant genes and one was a recessive. XA5 and XA13 are recessive resistant genes. XA21 is a dominant recessive gene. Where we progressed into this recipient price variety because it's a fine slender. Only we pulled the genes into this variety. And we made sure in the homozygous population the presence of all the three resistant genes. Only then we were.
Thank you, ma'am.
Yeah, to tell you more clearly, these were tested in both the seasons in 10 different locations. And we did half a dozen biological and technical replicas in our own greenhouse facility even before the national trials. Once we confirmed, then only we went to the national trials. And we need to do all the treatment, packaging, and then only we give away to the farm. And every batch we collect from different locations, we will batch test them. Every lot. So it's a lot of effort. And in 15 days, you will be working max. So I said 18 hours of work for 3 continuous years. You will not have time to relax. You have to be just running. That's it.
Thank you, ma'am.
Yes. Last question.
First of all, congratulations, ma'am, for your outstanding work in the Sangama program. It not only improved the crop quality, but also is really socially benefited. Also, congratulations. So as a biotechnology student, I'm a little bit more curious the disease bacterial life, right? So how did it act on the crop and what was your innovative solutions on it?
I didn't get the last one.
Repeat once again.
Hello.
Last one I didn't.
So I'm curious about the bacterial blight disease that act on the crop means created the disease and how was your innovative solutions on it?
Yes, so there are no pesticides for this. There is no chemical treatment for this, no pesticides. Okay? That is the reason we use the progression of resistant genes. It has no chemical control. And it's a very serious, devastating disease where a farmer can lose his whole crop. So that is the reason we chose the introgression of resistant genes into the rice recipients which we want. So we actually did it in two varieties. One is called PPT 5204 and the other is Triguna. Triguna is a little thick variety, but that also got released and it is going very well. But PPT 504, it is called now improved Sama Masuri. Further, our varieties will be called super improved Sama Masuri. So since there is no treatment, we introduce this resistance and we just drag it from the donor and put it into the recipient variety. And then we backcross to check for the presence of this resistant genes. Okay, they are not genetically modified. And what are the symptoms of this bacterial leaf blight? It begins from the tip of the leaf, graying and drying of the leaf, and will systematically come down the leaf and burn the whole tiller. So it spreads like if you light a fire, what will happen? So in the field, right? The whole field will look as if it is being burnt out because it's a grayish browning symptom which is caused by the pathogen called Xanthomonas oryzae bacteria oryzae. It's a xanthomonas and highly impious producing bacteria and it colonizes and spreads very heavily, very fast growth and grow across the leaf and you kill the whole leaf, it spread to the whole tiller and kill the whole bunch of leaves. And what will happen? So the grains will not get filled. They will all remain empty. So the farmer will not get any outcome, no seed will be and the crop is burned out.
Thank you, ma'am. It was amazing. Thank you.
Okay, thank you.
Thank you. Dear delegates and dear student friends, I believe in God, but I never see the God. But I see the God in some personalities like Dr. APJ Abdul Kalam, like Amar Kasai, Govind Singh Purana, M.S. Swaminathan. And I also see the God in the work of some scientists, researchers like Vishnu Priyana. That the great personalities do not require any introduction. But their introduction reflects with their behavior, with their words, and with their speech. Just we have experienced with the speech of Dr. Vishnu Priyana. And I just mentioned some research area and his work here. I just want to mention here. Mrs. M.R. Vishnu Priyana, ma'am, she had her earlier education in Hyderabad, Telangana state. Her specialized area of research are plant molecular biology and plant pathogen interactions. She obtained her PhD in biology and chemistry and MSc in biochemistry from Usmania University, Hyderabad. She has acquired her M.Tech biotechnology, Jawaharlal Technical University, Hyderabad, and PhD degree in botany from Usmania University, Hyderabad. She joined CCMB in the year of 1990, where she is currently chief scientist. Her research activities are focused on the understanding the mechanisms of the interaction between the plant and their pathogens, and understanding the biotic and abiotic stress in the plants. She is instrumental in the development of the improved Samba Unchari. Just we have heard about it. Bacterial blight disease resistant rice variety. She is the team manager of the CSIR 800 program on popularization of improved Samba Musri. In addition to her scientific activities, she is the head of the planning, monitoring, and evaluation at CCMB. She has visited several places in India to popularize the improved Samba Musri by making the farmers aware of the technological advantages in improving their income. This work has earned two institutional awards for CCMB. This is great achievement, students. From CSIR and DBT New Delhi. She has shown the quality technological development in CCMB at several mega science events, including India and international abroad. The International Science Festival, India International Trade Fair held in the New Delhi. Several interviews to print and electronic media were given while participating actively in the science popularization program. So this is a brief introduction of Dr. Vishnupriya ma'am, and I request Dr. Rahul sir to conclude this session with his concluding remarks.
Respected Chair, Dr. Gopalraj Patil sir, President Chhatrapati Shikshan Sansthan, Vice Principal Dr. S.M. Shinde sir, and Chief Patron Dr. Vishnupriya ma'am, Head Scientist CCMB, CSR Laboratory Hyderabad, Dr. ARDLS sir, then Dr. Salas sir, and all the dignitaries. So this is my great pleasure to be act as a chairman for this renowned speaker, Dr. Vishnupriya ma'am. So this is my pleasure. So, student friends, so Vishnupriya ma'am, this is a big idol for all of us because there is no limit for the science. I heard the biodata of ma'am. So she did biology, she studied chemistry, biology, then biotechnology. She works in all around the field, in COVID, in agriculture, in environment. So there is no limit for the science and no limit for the researchers. So ma'am has explained and covered all the topics like Shinde sir. So there is no topic for discussion further. So he covered almost all the topics of science, from medicine, agriculture, and environment. So very good take-home message given by the ma'am. So integrated biological science and for the well-being of the society. So we have to integrate all the sciences together and do a research in a way that it should be sustainable. There should not be a harmful impact on the society as well as the environment. So research should be integrated. So, so this is a very take-home message. And Madam also explained the several technologies which are ever growing, since CRISPR-Cas9 gene editing technology, then transgenics, then nanobiotechnology, bioinformatics, biosensors, artificial intelligence. So she has covered almost every technology, and she put very good examples of Samba. So this is the output. Even though we are doing several researches, but very few scientists come to the age and giving a good delivery to the society. So Samba Masuri is a very good example. So being agriculture, I know. So this is the just a few days back, our honorable Prime Ministers handed over these biopolitical reverse coordinations. So Samba Masuri, so there is a recipient parent and one is a donor parent, and we have made this several processes. It's a very big tough job because even though donor parent is there and recipient parent is there, but in every process 50% genome pool is going to transfer into next generation. So some undesirable genes are going to transfer into next generation, even though which are not desirable. So how to minimize this genetic background is a very big task. Because From donor parent, not only the single resistant gene is going, but along with this, some undesirable genes are going to the next generation. So, foreground selection and background selection is a very tough job by using different microsatellite markers. Madam has explained, they have used several markers, microsatellite markers to minimize the background of the donor parents and to enrich the backgrounds of the recipient parents with keeping a single resistant gene from the donor parent. So likewise, they have transferred three resistant genes and developed a variety showing a complete bacterial blight resistance to rice, and this is the satisfaction of the farmers. So this is a very novel and innovative works for the benefit of the society. So with this, again, I would like to summarize this topic. So ethics and policies, because the new technologies are going. So madam rightly pointed, ethics and policies. Because gene editing technology is there, transgenics is there, but not a single product is come out through transgenics, only beauty cotton. It is not edible, so that's why it is released for the farmers. Farmers going to cultivate since 2004. Only the beauty cottons, not edible crop being cultivated for the by the farmers. Because of the policies, because of the regulations. So very stringent regulations are there. And likewise, when several gene-edited crops are knocking the door for the commercial cultivation, but regulations are there. And so students should think in such a way that to frame our research, to design our strategies in such a way that so that it should be benefit to the society and it should not hamper the lives, microbes, environment, which is our resources. We have to preserve it for our next generation. So this is a take home message. I would like to conclude these sessions. Thank you very much.
Thank you, sir. Thank you all the dignitaries on the dais and off the dais. This is the end of the first technical session. Now we will going to take a break for the lunch.
So